Well, I thought I'd kick it off this evening by showing you a picture of this creature. And I would like to ask all of you if you can tell me what kind of a horrible monster this thing is. Pug? Give you a hint, we're going to be talking about biological evolution tonight. The evolution worldview would tell us that the Big Bang happened about 14 billion years ago. Some people say about 15 billion years ago. It was about 4.5 billion years ago that the Earth formed, so that's the age of the Earth. And about 3 billion years ago is when life somehow emerged from non-living chemicals in the primordial oceans. It was about 200,000 years ago that modern humans emerge. And the essence of biological evolution would be that natural selection plus mutations takes us from that single common ancestor to all of life on Earth. We contrast that to the biblical worldview, the Genesis worldview, and we would say that God created everything in 6 days about 6,000 years ago. And I know that a lot of people have some ideas that, well, maybe the days in Genesis were long periods of time. And they'll tend to want to fit millions of years into Genesis. But I'm going to make the argument next week that I don't think we have to. And that to attempt to do so would actually do some harm to the gospel. So we'll talk about that next week when I explain that. But if we do take Genesis seriously, the dinosaurs were created on day 6 with all the other land animals. And that's when, in the Garden of Eden, Adam and Eve rebelled and brought sin into the world for the first time and all of its consequences. It's about 1,600 years after creation that the flood came, Noah's flood. The geological column of rock layers is put down. Fossils are plants and animals that appear in the order in which they are buried. Okay? And after the flood effects trigger the Ice Age. There's one age, lasts about 700 years. People migrate to populate the earth after the incident at the Tower of Babel. We remember that, right? And of course, it's about 4,000 years after creation that Jesus comes to provide a rescue plan for that original fall into sin in the Garden of Eden. Got that? The biblical worldview. We got a lot of ground to cover tonight. Let's jump right into it with the origin of life. The origin of life. Scientists have been working for a long, long time now to try to understand how life could have emerged out of those primordial oceans. And right now, the answer to that is we really just don't know. We have no idea. Paul Davies, professor at Arizona State University and an evolutionist, has said nobody knows how a mixture of lifeless chemicals spontaneously organized themselves into the first living cell. And Andrew Null, professor of biology at Harvard, has said we don't really know how life originated on this planet. You see, the traditional formula has been simple. If we just have the right chemicals present, and we just have a chance, just a small, small chance that they could come together, we can do it. Why? Because we have 1.5 billion years according to the evolutionary timeline. But we now know that in order to have life, we need lots of giant molecules, proteins. Proteins are the basic components of every cell that has ever existed on Earth. And proteins are just these big molecules inside the cell that move around, and they make things happen, and they keep the cell functional, and they keep the cell alive. Now, proteins are made up of long chains of amino acids, kind of like this. And I'm going to borrow a couple of illustrations here from Dr. Stephen Meyer. But a protein is a long chain of amino acids. And amino acids in themselves are chemical compounds, and I drew a diagram on the board there. You get an idea what they look like. And to form a protein, we need to get amino acids to hook together in the right sequence, in the right orientation. And there are 20 different amino acids available to do this in nature. So they're kind of like the letters in the alphabet. Well, the amino acids combined, some proteins, the simplest ones, have about over 100 of them in a chain, and some of them have over 1,000. After the amino acids combine together, then it all gets folded up into a three-dimensional shape that is very important to the function of the protein. Now, as for the odds of this being able to happen by chance in the primordial ocean, take a look at this video clip.
As science reveals the intricate construction of proteins, origin of life researchers are confronted with a difficult question. Could amino acids floating in a primordial ocean have arranged themselves into protein molecules through purely random interactions?
Obviously, it is very difficult to imagine how in the primordial oceans the amino acids could organize themselves one after another into the right sequence to form even the simplest protein. And a good way to think about this is to think about the English language and how difficult it would be to string together a meaningful English sentence by randomly placing the letters one after another.
As an illustration, consider the difficulty of forming just one line of Shakespeare's play Hamlet by dropping Scrabble letters randomly onto a tabletop. Only one of 26 possible letters will work correctly at each of the 30 sites in the phrase. The probability of successfully assembling Shakespeare's classic line by pure chance is determined by computing 1 over 26 to the 30th power. The result is a staggering 1 chance in 2,810 trillion octillion. Yet while these odds are beyond comprehension, they seem almost reasonable when compared to the probability of randomly forming just one of the simplest protein molecules known to science. The smallest proteins in nature are comprised of about 100 amino acids. These chemical components must align in specific positions along a chain. Biologists have estimated that on a planet covered by a primordial soup, filled with complete sets of all 20 types of amino acids, the time necessary to construct a functional protein of 100 units would be roughly equivalent to the oldest estimated age of the universe, multiplied by 10 to the 60th power. But despite this inconceivable time frame, even if our protein somehow correctly self-assembled, the mystery of life's origin is still far from resolved.
Let's just say that against all odds, against everything we know about science, you do get one 100 amino acid long protein by chance. What do you have? Well, you have one protein. That's it. We don't have life because even the simplest cell requires over 600 proteins in order for it to be functional, in order for it to be alive. So mainstream scientists no longer think that life and the building blocks of life could originate by chance. They're now looking for a way to explain the building blocks and explain life's organization by some form of law or combination of laws that would allow that to happen. Chance is no longer an option.
All right, did you catch that last statement? They said that mainstream scientists no longer consider that the chance is an option. It's not on the table anymore. Instead, they're looking for some new law or set of laws of chemistry or physics that we haven't discovered yet that could allow for this to happen. Why? Well, Jonathan Sarfati, a PhD in physical chemistry, now with Creation Ministries International, has said, for example, the problem is the chemicals don't really even react in that way. Everything I've learned in real science, in real chemistry, shows that the reactions go in the opposite way from what's required for life to come from non-living chemicals. So This is huge. Our next big question for evolution would be, how can non-living chemicals organize themselves into the first self-reproducing organism? And this is huge because not only is chance no longer an option anymore in science, but it looks as though what we've learned from chemistry now is that it would prevent it from happening. Okay? By the way, we know that this is a big problem in science because we hear some scientists starting to say that, well, maybe life came from other planets and was seeded here on Earth. But can you see that that just puts the problem somewhere else? Because presumably, the same laws of chemistry and physics that operate here on Earth operate on the moon, in other parts of the solar system, in other parts of the universe. So it would be just as impossible for life to spontaneously generate somewhere else as it is here. Okay? Some evolutionists will say, well, they don't want to talk about it, the origin of life. They say, this has nothing to do with evolution. Evolution has everything to do with how, once we first have life, going from simple to complex. But if we're going to be honest about it, we would ask them, you know, well, this really has to do with the whole worldview. We're talking about a worldview, and the origin of life is critical to this whole thing. But it's a big problem. Okay. But we now know how those proteins form from those amino acids. Anybody want to know how? I knew you would. Proteins that are in the cell, that are already existing in the cell, if a protein needs to be built, other proteins will go to the DNA molecule. So let's talk about DNA a little bit. We now know that DNA is an extremely sophisticated digital code consisting of 4 letters. We call them A, T, C, and G. They're the initials of their chemical names. And they make up the rungs of the ladder of the double helix. Do you see the colors in the double helix? That's where they are. And they're arranged in a code. In a code. The very simplest cell that we know of in nature, the very simplest, contains about 580,000 DNA letters. That's enough letters if we're talking about the English language to fill a 200-page paperback. And if this neat little model I have here, this little toy, if this were a DNA double helix, even just for that simplest cell, this would need to be to scale about 3 miles long. And this cell is so simple that it can't even really survive on its own. It needs other cells to rely on nutrition. Now, the DNA contains the coding necessary to build proteins, the information. And what these other proteins will do, if they will go to the DNA and they will find the section that contains the code to build this protein. They will literally rip apart the DNA molecule and extract the information from there by creating a copy. They'll move the copy to another place in the cell where they will use it as a template to mechanically assemble these amino acids in the right sequence, in the right orientation, in the right order, until they've got the chain they need, and then they will fold it all up and they have a protein. Okay. The following video clip is an animation video of exactly that process. So take a look at how this works. It's called protein synthesis.
With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
Okay, so since the mechanics inside the cell, this is all very mechanical, isn't it? In order to build a protein, in order to build anything they need, first go to the DNA to get the information. Then the question of where first life came from really becomes a question of where did the DNA and the information in DNA come from? And as we focus in on this, we need to ask ourselves, is there any naturalistic reason for the letters in the DNA to be arranged in the positions that they are? And so the answer is no, there's not. Let's take a closer look at the DNA molecule and I'll show you what I mean. Here we have the double helix on the right side, okay, and over here, we just have it folded out flat. And you can see the sides of the ladder here and here in the gray. And along the sides of the ladder, we have these sugars and phosphates. And of course, the rungs of the ladder here is our code. A's and T's and C's and G's and so forth. Now, what's astonishing right here is that each one of the chemical bonds that attaches a letter to a sugar is all the same. And I would say that if this doesn't send shivers up and down our spines right now, it should. Because I would suggest we're looking at the fingerprints of God right here. I can illustrate what that is all about with these letters here. I don't know if you all can read the message. It's happy birthday, sweetheart. It was Jeannie, my wife's birthday last Thursday. So, you know, and if any of you would like to send some chocolates her way, anything that has chocolate, you know, you can bring them to me. And I'll be happy to forward them along. I'm just kidding. Don't do that. Chocolate is one of my big weaknesses, okay? So I love it too much. But you see here that each letter is positioned here with the same bond as the other. So there's no reason for them to be in a different location here. I can put them anywhere. The bonds are the same, like the DNA letters. Now, why is this so amazing? Because we know enough about the nature of things now in science to understand intelligence when we see it. You know, if we go out in the backyard and we look up in the plum tree and we see a nest, we instantly recognize that as not being the product of natural forces at work, right? Why? Because we're looking at an assembly of components that were put together by some intelligent agent for a higher purpose. The assembly itself has a higher purpose than any of the components themselves. And talk about an assembly made of components, what about these guys? Huh? I couldn't resist putting this picture in here. I love these things. My brother used to fly these. Or if we're walking down a trail in the Stanislaus National Forest in the Sierra Nevada and we run across a couple of arrowheads, we know that those are not the product of natural erosion, wind and rain. And by the way, neither are these guys, presidents' heads. We understand intelligence when we see it. Why? Because we know that nature left alone will give us one of two things. From chance, nature will give us complete randomness. Or, because of natural laws, nature will give us regular ordered patterns, like snowflakes or crystals, Na plus, Cl minus. We have a salt crystal, natural law. Or even sand dunes. Now, since DNA is a series of letters, kind of like a language, what would nature produce in DNA? Well, it would either give us complete randomness in the letters, or it would give us a regular ordered pattern. A, B, C, D, A, B, C, D, A, B, C, D. You see that? But that is not what we see in DNA. We don't see complete randomness, and we don't see a fairly simple regular ordered pattern. And this is the heart of the intelligent design argument. If any of you have ever heard of intelligent design, this is a group of highly educated professional scientists saying to the scientific community, time out. We're ringing the bell on this one. You got that? We want anybody to come to us and deny that this stuff in DNA is not evidence for intelligence. You see, they're saying, we're arguing from what we do know here, not from what we don't know. This is not a God of the gaps. Do you remember we talked about the God of the gaps last week? We have a gap in understanding of something we're looking at. We don't know what's going on, but that's okay. We know that God just did it. And however he did it, that's fine. Not really good science, really. We get criticism for that. But the intelligent design guys, team, are saying, this is not God of the gaps. There is no gap. In fact, someone wouldn't even be able to claim the nature of the gaps because the gap is filled in. All right? Now, not only this, but DNA contains huge amounts of information, not just the letters. So let's talk about information for a minute. You can all read the message in the sand? We know that the sand is not the information, don't we? And neither are the letters in the DNA the information for how to build a protein. Okay? Suppose I have a written report from any one of our students. And here it is. I'll pretend that this is a report on rattlesnakes that my daughter, Catherine, wrote last year. And I'm looking at this report, and I'm thinking, wow, who would have thought about, this is interesting stuff. Gee, I had no idea there were so many different kinds of rattlesnakes. It kind of gives me the willies. I wonder where this report came from. I wonder where this came from. Oh, I got it. Doggone it. I know where this report came from. I recognize the paper. I had this paper in the upper desk drawer, and I went and I left one of those pens on top of the paper again that leaks. This has happened before, and overnight the pen leaked and spilled ink all over the page, and here it is, doggone it. No. We all know that that's silly, right? No. If we look at the DNA letters here, where did that message come from? Thank you. Wow. It came from me. There is no known law of physics, gang, that tells us that we can get information from nothing. And I would say that everybody in the world, every rational person understands that information requires an intelligent source. So, next big question we have for evolution is, where does all the information in DNA come from? And this is huge because we don't have the gap. We can see the letters in DNA, and there is no naturalistic reason for them to be arranged the way that they are. Okay? And we know that information requires an intelligent source. All right, how are you doing? Are you following me? Are you with me? Because I know we're getting into some depth here. You know, we're covering this stuff pretty fast and it's pretty deep, but I had a good feeling about this group tonight. You guys are doing great. So let's keep on going. Now, before I zoom out of the cell, and I'm going to do that in a minute, I want to talk about something else that's going on inside the cell that is very interesting. Let's go back to that simple cell one more time. It's got 580,000 DNA letters. It also has about 300 enzymes. And enzymes are just other big giant molecules, proteins. They're a certain kind of protein. And it's the job of the enzymes to extract the information from the DNA. Okay? They speed up the process. If they didn't do that, it would never happen. They're catalysts. Enzymes are catalysts. Enzymes extract the information we need, but the information to build an enzyme is located where? In the DNA. So when we look at the cell, we're starting to see a chicken and egg problem, aren't we? But it gets a little more complicated than that. In order for the enzymes to do their job, to get that information out, they need energy. And they get the energy they need from this little motor called the ATP synthase motor. This is an assembly of several different proteins, and it provides the energy for the enzymes to go get information. But part of the problem is that the information to build an ATP synthase motor is located where? In the DNA. So what we're really looking at is a vicious three-way circle. And creation scientists have a term for this. They call it irreducible complexity, and the mousetrap is the classic example. See, you have a number of different components that must be present all at the same time, or else it doesn't work at all. You take away the wood in the mousetrap, and all you have is a little pile of useless parts, don't you? You take away the spring, you don't have anything that functions. And this is a big problem for evolution, because evolution, by definition, says that we go step by tiny step from simple to more complex in increasing value. And if we ever get to a point where we need a number of different items to suddenly show up on the scene in parallel at the same time, that is more akin to a miracle, and that is not in harmony with evolutionary theory. Big problem for evolution. By the way, that little ATP synthase motor is in itself irreducibly complex. Take a look at this video.
Most cellular processes, including things like DNA replication and protein synthesis, are powered by a molecule called ATP. And the ATP synthase motor is one of the wonders of the universe. It's a tiny electric motor required by all life. This rotary motor uses protons to generate spin and then uses that to join ADP molecules to phosphate molecules, creating ATP, or adenosine triphosphate. All life is based on ATP. It's called the energy currency of the cell, and your body produces its own weight of ATP every day. And that motor works at nearly 100% efficiency. ATP is a complex and reactive molecule not found in abiotic systems. Yet without ATP, life could not exist. How did life decide to use that molecule for energy, and where did it get it before it could produce it?
You know, in fact, the cell, we're finding out, science is finding out that it is just filled with irreducible complexity. These great big proteins that get assembled when they're built, when some of them need to be transported to a different part of the cell to start doing their job and going to work, how do they get there? Well, they take the postal delivery system, of course. Let me introduce to you the cell's mailman, the kinesin linear motor. Take a look at this video.
Cells are full of specialized components that perform functions vital to their existence. But how do these components get to the right locations inside the cell to perform their functions? For larger components, a transportation system is needed. Meet the kinesin. Masterpieces of microengineering, kinesins are miniature motorized machines that carry cargo from one part of the cell to another, walking along self-assembling highways called microtubules. Known as the workhorses of the cell, kinesins have 2 feet, or globular heads, that literally walk 1 foot over another along the microtubule, pulling their cargo to its destination. Each foot possesses 2 special locations called binding sites that interact with other molecules. One site attaches to the microtubule and the other binds with ATP, the energy molecule of the cell. When one foot binds with ATP and uses its energy, the foot flips over, resulting in the walking motion. Each foot has a short neck which is connected to a strand of a long coiled stalk. At the end of the stalk is a fan-shaped tail which holds tightly to the cargo being transported. Kinesins can carry cargo that are many times their own size. Sometimes a kinesin is in danger of getting stuck on the microtubule highway because of blockages caused by other cellular components. To get around such obstacles, multiple motor proteins may be used to carry a single piece of cargo, together providing enough force to break free. Kinesins typically walk away from the center of the cell and toward the cell's periphery. The kinesin's 2 feet work together efficiently, with 1 foot holding fast to the microtubule, while the other releases itself and takes a step forward. This coordinated stepwise movement allows kinesin motors to walk as many as 100 steps per second, moving about 8 nanometers with each step. When not carrying cargo, kinesins can shift to energy-saving mode to conserve fuel until their next job. The kinesin plays a vital role in many cellular processes, not just transporting materials, but also aiding cell replication. The walking kinesin molecular machine, another example of intelligent design.
Charles Darwin once said, "If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down." You know, we used to point to the eye. Look how complex the eye is, or the wing. But now we have the cell, the basic building block of all of life on Earth, showing incredible, undeniable, irreducible complexity. And according to Darwin, the theory would break down because of this. But in Charles Darwin's defense, he knew nothing about any of this. In Darwin's day, the cell was considered, quote, a simple lump of albuminous carbon. It means that they knew that the cell was kind of filled with clear fluid, but like the white of an egg, if you heated it up or cooked it, it would turn white. And that was about the extent of their knowledge of the cell in his day. So how are you doing? Are you following me? Yes. Good. All right, you're doing great. Now, let's zoom out of the cell and start talking about evolution from simple to complex. And when we do that, I'm going to go back to the DNA molecule. That simplest cell, we have that there. We know that the human genome has 3 billion DNA letters. That's enough to fill 5,000 200-page books. And if I look at this model again, if this were some kind of a scale model, and if this was a human DNA, this little plastic chain would have to reach 15,000 miles. Okay? Now it turns out that DNA, it doesn't just contain the information on how to build proteins, it basically contains the information about how a given organism is going to develop body structures, like blood, nerves, muscle, bone, hearts, lungs, feathers, wings. Now, if evolution were true, it needs to give us some mechanism whereby we get all the additional information required along the way, where we get the information from nothing. In other words, how did we get the information, the additional information required to go from a single-celled creature to, say, a horse? Here's a true story. When Darwin visited the Galapagos Islands, pardon me, he was studying a population of finches. And he noticed that over time, the beaks in his finches could vary. In drought years, when the seeds that they ate were extra hard and tough and kind of large, only the birds that had big, strong beaks tended to do well. And Darwin figured, well, Darwinism became the idea that, hey, maybe if we see that kind of adaptation in animals and we let them accumulate over plenty of time, we could get evolution from one kind of animal to the next. This became known as Darwinism. He termed this descent with modification. And the tree of life model, whereby all of life on Earth is descended from one single common ancestor, was born. But, still true story, at the time that Charles Darwin published this theory in Origin of Species, Gregor Mendel, who is considered the founder of the science of genetics, was just getting started with his experiments that would show us how we really inherit traits from our parents. And this is fairly well known today. It's called Mendelian genetics. And we all understand this, but let's do a little bit of a review. Suppose we have a breeding pair of dogs. And each of those dogs happens to have the genetic information inside it that codes for long fur. We'll call it the L gene. They also happen to have the genetic information that codes for short fur. We'll call that the S gene. And we know that if a dog has those genes in combination, they might have medium fur. We also know that if those dogs had pups, those pups would inherit any possible combination of those genes, wouldn't they? Some of them would be like their parents. Just never mind the red X's for now, they'll be apparent in a second. Some of them would be like their parents and get both genes in medium fur. Some of them would get only the short fur gene in combination and wind up with short fur. Do you see that? Some of them would wind up with only the long fur gene, and they would get long fur. Now, what happens if this growing family of dogs moves up to northern Canada where it gets very cold? Any of the dogs that have short fur or medium fur are going to get very uncomfortable, and if they're smart, they're moving down to Cancun, aren't they? Very quickly, within just a couple of generations, we have a population of dogs that only has long fur. What happened? Are we evolving something new with something new with long fur? No. And scientists began to understand that natural selection only operates on available information, and if anything, as animals adapt in different habitats and environmental conditions, they're going to lose information as they adapt. They saw that Darwin was wrong. His finches did not develop the new information for big beaks. They had it all along. If anything, they lost the information for the other kind of beaks. So those who believed in evolution understood that they still needed to find a mechanism that would increase information. And in the 1930s and 40s, it became these guys. Oh, I forgot to show our last row of dogs up in Canada. But in the 1930s and 40s, it became these guys. And who can tell me who they are? Yeah, that's what they are. They're the X-Men. They're mutations. The new Darwinism became the idea that, hey, look, every once in a while a mutation's going to come along. Natural selection will keep the good and get rid of the bad, and we can go from evolution, one kind of animal to the other that way. Now, I do not mean to mock Darwinism with this cartoon. I really don't. But I just want to point out that even though mutations can make for great science fiction, and I like science fiction, we now know that mutations do not create any new information either. Mutations are just copy mistakes as we copy our DNA from one generation to the next. And since DNA, again, is a written code with letters, the written English language is a good illustration as to how this works. Let's take a look. Let's start with that sentence we saw in that video clip, to be or not to be, that is the question. We go to copy that, a mutation comes along and we swap a letter. Do you see it? Or another kind comes along and we'll swap a word. Or mutations can even delete a phrase or duplicate a phrase, but can you see that we're not adding any new information here? If anything, mutations will corrupt the genetic information in DNA over time. Now, most mutations are neutral. They don't cause us any harm. Some cause diseases like sickle cell anemia or cystic fibrosis or 1,000 other diseases. But interestingly, once in a while there can be a beneficial mutation. The classic example is beetles on a windy island. Every time they get up to fly, they get blown out to sea and die. But a mutation comes along and corrupts the genetic information responsible for their wings developing properly. They stay on the ground and they do well, and that's okay, isn't it? But they're starting to look different from their cousins on the mainland whom they got separated from, aren't they? Are we evolving something new with more information? No. We still have a beetle. We still have a beetle. Bacteria resistance to antibiotics is another example of a mutation. Mutation has come along and corrupted the genetic information that is responsible for that bacteria being sensitive to that poison. Follow that? It has broken that bacteria's ability and transfer mechanism to move that poison molecule in through the cell wall. Can't do it. We see animals out in the wild that have different colored coats, like our tigers on the first slide. Mutations can be responsible for variations in coat colors. Some of those can be beneficial, and we see them in nature because they've survived. Interestingly, there are a lot of mutations in the domestic dog breeds. We take a look at Rosie again. Dogs like pugs that have flat faces, that's a result of a mutation that came along that has corrupted the genetic information that they have responsible for their muzzles growing out properly. But what I find interesting is that same mutation in the pug did not affect the growth of the skin, did it? That's why, as they grow older, they have a lot of extra skin and all those cute wrinkles because they got enough skin for the properly developed muzzle. Dogs with short, stubby legs like dachshunds, well, that's a result of a mutation that has damaged the information responsible for their legs growing out properly. Okay? Dr. Lee Spentner, an expert in this area, wrote in this book, he said, "In all the reading I've done in the life sciences literature, I've never found a mutation that added information." Let's stop for a minute there. This is a world authority on mutations. That means that if any one of us wanted to go out and take on a personal project for a whole year and canvass all of science, we would never find a mutation that added information. He goes on to say, "Information cannot be built up by mutations that lose it. A business can't make money by losing it a little at a time." Okay? Sometimes I illustrate this with this little Lego boat. You see this? This is a Lego model. And the instructions to build this model are here, this. And I know that Lego instructions only have pictures, but let's just pretend they have sentences and words and letters. But these are the instructions on how to build a boat. Now we have a little exercise to do. Here is our challenge. I want us to evolve these instructions so that we can build one of these, our flying creature. Okay, we're going to go from an aquatic creature to a flying creature. And the instructions for the Millennium Falcon, I didn't bring it in because it's too sensitive, it's too hard to carry, but it's awesome. It's big and detailed, and my son TJ built it. The instructions for the Millennium Falcon look like this. They're a lot more complicated, aren't they? All right. Now, to evolve these instructions into those, we may delete some instructions. We can swap some letters, words, or phrases. We can duplicate or delete some letters, words, or phrases, but we don't have to worry about it very much. We have all the time in the world. You see how that works? All right. Let's talk about the creation model. We don't believe in a tree, we believe in an orchard. Because in Genesis 1, it says, each according to their kind. It tells us that 10 times in Genesis 1. We believe that God created original animal kinds with vast amounts of genetic information because He would want their descendants to be able to adapt to different habitats and environmental conditions around the world. It's estimated that there were about 8,000 different animal and bird kinds that were saved on the ark. And all of the land animals and birds on earth today would be descended from those original kinds. Let's take a look at how this works. Let's look at our dogs, for example. Okay? It's estimated now that all the dogs on earth are descended from a single pair of dogs, and recent genetic studies would suggest that that's true. God creates a pair of dogs. They start a family, they grow, they split up and they migrate, and they maybe start to adapt according to natural selection. Along comes the flood. The basic animal kinds are saved on the ark. God supernaturally calls the animals to get on the ark. Noah didn't have to go out and get them. Do you remember that? That pair of dogs gets off the ark, and they start a family, and they grow, they split up, they migrate to different areas. Some go to northern Canada, some go to Cancun, and what do we have today? We have wolves, coyotes, dingoes, jackals, foxes, not to mention all the domestic dog breeds. But what happened? Have we evolved different kinds of dogs along the way? No. This is natural selection in action, where if anything, those dogs lost genetic information as they adapted to different areas. Let's take a look at the ark for a minute, by the way. Is this what the ark looked like? This is an outstanding piece of art done by my daughter Suzanne, and her sister Catherine did one just like it. And I used to make these in Sunday school, right? Because we should celebrate the story of the flood, and we do celebrate it. But this is not what the ark looked like. The ark was an ocean liner-sized vessel. The dimensions are given to us in Genesis. It was 450 feet long. Isn't that a 45-story building? Leaned down on its side. It was 75 feet high and 45 feet wide. And if you do the math, an ark like that could fit about 125,000 animals the size of a sheep. Now, if we're going to save 8,000 animal kinds, tell me, somebody, how many animals need to go on the ark? 16,000. Now, some were kind of special, and God required a few more, but that leaves plenty of extra room for food and air circulation and so forth. As a matter of fact, studies would tell us that estimates are the median size of the animals on the ark were about the size of a small rat. And only about 11% of them much bigger than that. Now, as for what the ark might have really looked like, let's take a look at this video clip animation. Now, I know that doesn't show us very much, but I just love that video clip. I wanted to include it. The ark. Let's go back to our model. And look at the primates. Now, I don't know how many different monkey or ape kinds would have been saved on the ark, but same thing. They get off the ark and they split up. They begin to adapt and diversify, but many of them don't make it, do they? Can you see that? We know that from bones that we find. Some of them alleged to be part human, by the way. Let's look at the dinosaurs. If we take Genesis seriously, then the dinosaurs would have been created on day 6 with all the other land animals. It's estimated that there were about 30 different dinosaur kinds that were saved on the ark. They were about the average size of a sheep. Most of them were small, and if any of them happened to be big, they could have been brought on board as what? How would you bring them on board? Juveniles. Yeah. In fact, the biggest dinosaur egg that we know of is about this big, right here. Pretty big. Same thing, they get off the ark, they begin to migrate, adapt, and diversify, but none of them make it. Various theories why. But can you see that the creation model is in perfect harmony with natural selection and mutations? On the other hand, natural selection and mutations can't support the tree of life model because the tree of life model requires an increase in information over time. We don't need an increase in information over time here. Okay. When I spoke to the high schoolers about all this, I asked them if any of them recognize this book, and probably half of them raised their hands. This is the biology book used at Santa Teresa High School. I borrowed it from the principal. And I wanted to point out to them that textbooks can be very confusing about all this stuff. In fact, starting right off the bat, we have a long discussion in the book about evolution in chapter 1. Page 8, it says, scientists suggest that natural selection is the most important driving force in evolution. Now let's stop there for a minute. Well, natural selection is responsible for getting us all the animals on Earth today from the original kinds, but it kind of depends on what they mean by evolution here, doesn't it? So we got to be careful. It goes on to say this descent with modification. All right, so they're using Darwin's term. They're talking about evolution across animal kinds. Is responsible for the multitude of life forms we see today. This just isn't true because Darwin's descent with modification is not responsible for all the animals that we have on Earth today. So it's very important that our students understand this when they see this in their books. And every example in the book that was given for evolution was nothing but an excellent example for natural selection. There is no new information created. But there is an unspoken and silent assumption in the book that adaptations, according to natural selection, can accumulate over time and cause big changes. But there's no evidence for that. So, for the tree of life model, I would repeat the question. Where does all the information come from? There is no good answer for that. And, you know, amazing to me, evolutionists will still hang on to natural selection and mutations, even though scientists that are involved in the field must understand that that can't be true, but they insist on it because it's all they have. But the idea is bankrupt today. It's bankrupt. Okay. Done good? Let's look at our summary for tonight. We're not over quite yet. We need another minute. All scientists today agree that life cannot start just by chance. In fact, the laws of chemistry would seem to prevent it from happening. Life is built by first extracting information from DNA, and there is no naturalistic explanation for the arrangement of DNA letters or the origin of the information. Irreducible complex machinery inside the cell is powerful evidence for design and is something that Darwin just knew nothing about. Neither natural selection or mutations increase information over time. Natural selection works with existing information, and mutations corrupt it. There is no evidence to support the tree of life model. On the other hand, the creation model is in harmony with natural selection and mutations. Next week, we're going to get into the age of the earth. We're going to look at rock dating methods and how those work. We're going to look at the fossil record and what kind of evidence that really is for us. And we're going to wind up with a discussion to cap it all off with why all of this matters. And just before we close, I want to wrap it up with a couple of quotes, if I can. Francis Crick, the co-discoverer of DNA, won a Nobel Prize for it, has said, "Biologists must constantly keep in mind that what they see was not designed, but it was evolved." And Richard Dawkins, an author and a real well-known atheist, Oxford University, has said, "Biology is the study of complicated things that give the appearance of having been designed for a purpose." And I have a comment about that. Maybe they were designed for a purpose. Let's pray. Heavenly Father, thank you for all of these exciting things that we're learning in science these days. We thank you that we can see that it just aligns so well with what we read in Genesis. Lord, I ask that you take all these folks home safely tonight, bring them back next week safely as well. We thank you for all these things in Jesus' name, amen. God bless you and good night.